Inside a sterile laboratory at Harvard Medical School, the air is thick with the hum of processors. Zinan Zhou sits motionless, his eyes fixed on a screen of raw sequencing data. For decades, the world of medicine has viewed the giants of cognitive decay as isolated islands. Amyotrophic lateral sclerosis, frontotemporal dementia, and Alzheimer's were treated as separate tragedies, divided by their unique protein signatures. One was defined by TDP, 4-3 clumps, the other by tau tangles. But Joe is looking for a ghost in the machine of the human brain, searching for a secret that links these disparate paths of suffering. He selects a specific sample from a patient with C9orf72 type ALS. The team initiates single-cell whole genome sequencing on 469 individual neurons. It is a massive undertaking, peeling back the layers of biological code.
As the results flicker onto the monitor, a chaotic landscape of somatic mutations emerges. In these diseased cells, small insertions and deletions, known as indels, number over 1,000. It is a biological time bomb. This level of genetic damage is equivalent to centuries of natural aging compressed into a single, tragic lifetime. While healthy neurons remain stable, these cells are literally tearing themselves apart.
Joe filters the sea of data, searching for a signature, a fingerprint left by the culprit. He finds it, a distinct pattern of 2 base pair deletions. It is a perfect match. The culprit is top1,
the DNA topoisomerase enzyme. Experiments soon confirm a horrifying reality. Toxic top1. DNA covalent complexes are clogging the neurons like grit in a delicate engine. These enzymes were meant to assist in the transcription of life, but they have turned against their hosts. They are carving permanent, jagged scars into the very genetic code they were designed to protect.
Joe moves to the next slide, mapping the devastation across the brain's geography. The cortex is ravaged, yet the cerebellum remains eerily untouched. This damage is not random. It is a calculated assault dictated by the high metabolic demands of specific neuronal regions. He leans back in his chair as the implications settle. For years, researchers chased different proteins for different diseases, lost in the symptoms. But now, the common denominator is exposed. Whether it is ALS, FTD, or Alzheimer's, the underlying engine of destruction is the same. top1. Mediated genomic instability. The ghost in the machine finally has a name.
The screen glows with the highlighted target. By intercepting the top1 pathway, the entire landscape of neurology could shift. No longer are we looking at a dozen different wars, but one central battlefield. This discovery bridges the gap between these devastating conditions. It offers a unified path toward treatment where once there were only disparate, dead-end roads. The data on the screen is no longer just numbers. It is a blueprint for a new generation of medicine, a way to stop the clock on the biological time bomb before it's too late.
Zinan Zhou looks away from the screen, the sterile lab suddenly feeling full of possibility. The search for the ghost is over, and the fight for the future has begun. One target, one strategy, and finally, one hope for millions. The engine of decay has been found. And now, we can learn how to turn it off.